We don't need music. No music. No music.
Hey,
Get rid of the
everybody.
music.
Welcome to Q&A with A&V. Here's, I'm Vincent Racaniello, and here is Amy Rosenfeld. Hello, Amy.
Hello, Vincent. No music.
Why not? What's wrong with
No music? No music.
Why?
No music, and especially not music that is TWIV music. No music.
My goodness. Hello, everybody. How's our audio?
We are different.
Folks, before we start, tell us how the audio is. We'd like to know. Does it sound okay? Is Amy too loud? Is she too low? Let's go. Hello from Brazil. Amy is insufferable. Thank you for giving us this weekly gift, Sweden. Hello, folks.
If I'm so insufferable, why is he listening?
Just to bug you. Frank likes the music. Audio is good. Okay.
No
Here
music.
we go. Here we go. But I think that in live streams, people usually use music.
I don't care. No music.
Well, Amy said it. She doesn't want music, folks. May I suggest you use Slido for the questions instead of Super Chat. I don't know what that is, but I'll look into it, folks.
Okay, well, look at Slido.
Hello, Seattle, Chicago, UWS.
Upper West Side.
Wow, that's good, Amy. I wouldn't have gotten that.
You are not
here
in
we
New
go
York
with our first question. Oh, look at this, a high school student. Hello, Amelia, welcome.
It's great.
Second best show on YouTube outside Mossy Warrior Clan. Thank you.
Do you watch that? I don't know what it is.
I don't know what it is, but thank you for that. Being the second best is good. Okay,
well,
Claudia
I don't know how you're going to compete with something that's called Mossy Warrior Clan. Sounds like there's some scantily clothed women in that.
Is it mutually exclusive for a virus to be highly infectious and highly deadly? So highly infectious, do you mean by that highly transmissible from person to person and highly deadly? So here's the thing. Rabies virus is highly deadly, but it's not very transmissible from person to person. Ebola virus is very deadly, but not very transmissible. So it's not a rule, but I don't see it yet. I think there has to be some balance, frankly. Warrior.
I don't know.
Okay.
This guy understands. Author wants music and told me to go.
Questella understands you not wanting the TWIV theme. Yes, Questella, but she doesn't want any music, okay? That's fine. Here we go. This is a nice teaching moment. Can you give a short summary of the RNA test and what the terminology means? You want to go at this, Amy? This is your shtick.
No, I don't want to go at it. I was told to go.
No, you're not going to listen to people. People want you here. There are a few people who tell you to go. There are a few people that tell me to go. Don't worry about it. All right, RT-PCR, reverse transcriptase polymerase chain reaction. You want to know how much RNA is present. You extract nucleic acid, then you convert the RNA to DNA with reverse transcriptase. It's an enzyme. You can buy it, or you can borrow some from Amy. She has it.
I do.
And then you add primers that are going to amplify a small piece. You put it in a PCR machine, which you can also get from Amy. She has a couple.
No, I don't have the special one.
You go through 40 cycles,
have the special one.
And you see if you get a signal. And the cycle where you get a signal, that's called the cycle threshold. NAT, nucleic acid amplification test. That's kind of a catch-all for all these tests. And LAMP is a different kind of amplification using a different polymerase entirely. Right, Amy?
Yeah, it's isothermal.
What does it stand for?
Remember? I don't remember. Hold on a second.
Oh, loop-mediated isothermal amplification.
Yeah, it's isothermal, yeah.
So they all amplify nucleic acid.
They're all basically the same thing.
Yeah, and so I think
they've just enhanced the technology, and LAMP is very fast. And isothermal makes it go at the same temperature, at room temperature or ambient temperature. So you don't have to schlep around a machine, which makes it ideal for when you go to Africa and other countries that don't have such technology as we do, or wealth as we do.
Why don't mRNA vaccines need adjuvants? Actually, they have a built-in adjuvant. The lipid nanoparticle probably serves as an adjuvant, right, Amy?
I would think so. Yes, I would think that they just don't have the traditional adjuvants.
Yeah, like aluminum hydroxide or
But I
squalene,
also
whatever.
right, but those are with proteins, right? Those are so this you're programming the cell to make that thing that's going to elicit the immune response. You're not giving it to the cell for the eliciting the immune response. You know, it's one step
Yeah,
beforehand. So I don't think an adjuvant is necessary. You need something to bring it into the cell more efficiently. That's not what an adjuvant does. The rest is left up to the cell.
All right. We have two questions on the recent California variant. Question: hype or real California variant? And Frank says, what do you think of the UC researchers saying the devil has arrived for the California variant?
Charles Hsu.
I would not listen to Charles Hsu. Bottom line. Charles Hsu does not understand virology. And as Nels said today on our live stream, don't listen to hearsay. Wait for the manuscript and we'll look at it, because this is not even a preprint. Okay? I don't believe it. Anyway, the numbers are going down, folks. It's all good. What happens if you get mild or symptomatic COVID and get vaccinated?
You should get vaccinated. And I believe that there's a bunch of preprints on bioarchives that talks about when people who got COVID and getting vaccinated and their responses. And I believe that they conclude from various papers that you only need one dose and that you should get vaccinated. And it boosts your immune response.
You should wait till you recover, though, right, Amy?
Yeah. Yeah. Well, you don't want to contaminate the people who are vaccinating you, right? More important.
Yep. In Brazil, they will vaccinate 100% of the population of a small town next month, around 30,000 people. That's great. Good real world data can emerge from that. It could if they collect it. I hope they do, and they have good science there. So I look forward to seeing that.
Sounds great.
Barry says, seems like thick sputum might affect droplet size, float time, and transmissibility. Does dry versus wet cough correlate with R0? So here's the thing, Barry. You don't need to cough or sneeze to expel droplets. Talking alone will expel droplets. So I'm not sure how thick sputum will affect that. Mayo says measles and chickenpox, dry cough. I don't think a cough see, for flu, coughing and sneezing doesn't matter. Speaking is enough to transmit flu. Are the viral genetics of what causes wet versus dry cough understood? No. I've never seen a paper on it. Have you, Amy?
No, I'm not clear how you would do that.
If you test positive for COVID-19, should you refrain from testing for 90 days? No.
What do you mean? I don't understand the question. You should, if you test positive, you can get tested 10 days later and see if you're negative. I don't really know.
We just got a super
where
chat
90
from
days came.
Post Polio in support of Amy's no music. Okay. Fine.
You got a what?
A super chat from Post Polio News in support of Amy's no music.
That's funny.
So, no, I was tested positive months ago, and they said, come in next week and get another test and to make sure it's a negative. And that's what I did. So I don't see the issue.
But that's not, I don't think that that's what they're talking about. I think they're saying if you test positive and you actually have the virus, should you refrain from being tested for 90 days? No, the virus is cleared within 7 days. So you should get tested at days 10. But, you know, I'm not sure why you would continuously get tested, but I thought the policy was, or the CDC guideline was, you got tested until you were positive, and then you quarantined or isolated, whatever the right word is.
Mm-hmm.
And then you just, as long as you had no symptoms after 10 days, it was fine.
Both California and New York allegedly have variants of concern that are more transmissible and more deadly. Why do I not buy this? Because you're
it's wrong.
smart. It's wrong, Jeffrey, and you got it. It's all hyped by the media, who find people that say what they want. But I can find you as many people who will say they're not more transmissible and more deadly. So just don't buy it. Listen to what we say. UK has revealed its roadmap. Shots for all the most vulnerable. Open up everything and let the virus rip. What do you think, Amy?
Not the plan that I would have gone with, but I am not Boris Johnson, so I don't get to make those decisions.
I think it's good to give shots to the most vulnerable, but to open up and let it rip is really dumb, in my opinion. But nobody asks me. I don't direct any policy, neither do they ask Amy. So that's why we talk here on Twiv and Livestream.
Yeah.
Your views on using antigen tests as a way to create herd effect, as they test for contagiousness and are 99% accurate for that purpose. I guess this is the MINA test, right, Amy?
I don't know if it's the MINA test. It's the MINA idea, right?
That's right. Sorry,
But,
it's the MINA idea.
So you test, so if you test for antigens, it tells you that that you potentially were infected. I don't know how that's going to create herd immunity. I would think you would need to do antibody tests.
Right.
And I'm not sure how antigen tests are going to test for or give you any information on contagiousness. I don't see the connection. It doesn't tell you anything about the virus. It just tells you you produced a viral protein.
Yeah.
So I don't know
Right.
what the connection is. It's not clear to me. It seems to be a jump.
Arthur Garzon gives us a super chat. And we will get to those in the chat. But thank you, Arthur.
Yeah, in support of the music. That's hysterical.
So I'm getting for and against the music, right, Amy?
Yeah.
Amy, if we had music you liked, would that matter?
I might go for that.
We got Frank Peace super chat.
I might go for that. I might go for that if I got to pick the music.
All right.
Then we can discuss with Jonathan, my music consultant.
Okay.
My lifestyle music consultant. Okay. We'll discuss with Jonathan, the musician.
No Prince.
Don't worry, he doesn't like Prince. Okay. He's an Annie-Lennox guy.
Susan, pair character lifting some weights. Keep it up, Susan. Right. Thank you, Susan K. All right, answer this question.
A zoonotic virus is geographically limited until it encounters humans. Zoonotic viruses zoonotic viruses can traffic between animals, and they can move from animal to animal without encountering a human until the very end.
Yeah, they're not necessarily limited. No, not at all.
Well, they're not limited at all. I mean, I think that was part of Ian and Peter Daszak's paper that you guys did on Twiv, and Lympha's paper and stuff. And then if you look at, all the history of Hanta and whatever Four Corners virus here, they go from animal to animal.
Mm-hmm. I think
if you look at flu, right? You can go from bird to pig, and then that pig, then go back to a bird, and then the bird flies 1,000 miles and infects another pig.
Mm-hmm.
So,
do you want to discuss the ethics of challenge trials? I'm fine with it and would gladly volunteer for such a trial.
No, I don't want to discuss the ethics. I'm not a bioethics person. I'm not a lawyer. I'm not an MD. I don't.
Thank you, Jeffrey, for your super chat. Jeffrey Millard. I don't want to discuss ethics, but I think if you are challenging unvaccinated people, that's a problem, especially with SARS-CoV-2, because it could kill you. So I don't see why you would want to do that.
Even more importantly, I think that there is a significant number of people who are in what we have defined kind of irresponsibly as age groups that are less affected because they don't die. But in reality, they may be even more affected because they are the age groups that define long-term COVID. So I don't know that I would take 18-year-olds, give them virus, and then have the potential for them to develop long-term COVID.
Thank you, Jeffrey, for your super
What do you
chat.
think about that?
Yeah, I agree with you.
So I think that it's a mistake that we have made by saying that there's this like age cutoff because you don't die. But we all know, you and I know several people who have long-term COVID who are their lives have been uprooted from it, right?
Yes,
and
completely.
As a physician or a bioethics person, I'm not clear how I could rationalize that in my mind as this was appropriate. So therefore, that's why I didn't become an MD. I don't want that responsibility. And I'm surprised maybe this Adam person will reassess and say I'm fine with it when he realizes the potential of him developing long-term COVID is not so small.
What do you think about the California Health Department saying it would not recommend a single dose J&J to over 65 and those with comorbidities? 71% is not effective, he says. I guess this came out today, right, Amy?
I was unaware of it. Sure.
The FDA advisory committee said it is safe and efficacious, and 69%, 71. I think this is fine. I don't see why this person would not recommend it. I don't agree.
I don't agree either.
Oh, H5N8 avian influenza, another virus taking our attention away from SARS-CoV-2.
Wait a minute. It's not polio? I'm sorry. I do not process anything but polio as the other virus in the world. Sorry.
Thank you, Tom. $10 super chat to help with our studio. Thank you. We're working on it. I think the H5... Okay, so Susan, thank you for the super chat. Lovely. Your folks are lovely. This has now apparently gone from birds to people in Russia. We've seen that before with other kinds of avian influenza. The question now is whether it spreads in people. I doubt it will.
To person.
Yeah, but H5 influenza, avian influenza, has been sporadically jumping into people for 50 years, and it has never gone person to person. So I think it's unlikely, but we'll see.
Yes, we shall see.
All right, John says J&J trials show 72% efficacy, which increases to 85 after 28 days. Amy has said it takes months to gain full immunity. Does the 85% represent antibody immunity? Could 85% increase given more time?
I think probably it will go up given more time.
It's hard to know if it's antibody or T cell or both, right?
Yeah, I don't know what it is. But we only test for antibodies. We don't test for T cells and stuff.
Yeah, that's right.
So,
but I would guess it would go up.
Yeah, I think it goes up.
And that's why the people who say, well, one dose is enough, you can't really say that because those people got a second dose, and so you never go past 28 days, and you don't know if it would decline or not. So, yeah, this is a problem.
Well, we decided they decided that they needed a boost. And as I said, from my point of view or my perspective as an outsider, it seems to me to be so that you remember to go back. Seems to be about that versus the science. Because if it was the science, then the trials would be a lot longer, right? Because you would
Yes.
have to pace the boost out to see when it was best.
Wesley says, Moderna dose 2 kicked my butt. Do I really need to do that again for every variant of concern?
No, because there seems to be, they only test against monoclonals. They don't really test against polyclonal serum, and it doesn't seem to be the fourfold decrease against polyclonal serum. But I did read today that Moderna has a variant, a second generation vaccine ready for testing, ready to go.
Yeah, the FDA has said you don't need to do a phase 1. You can just make it and do some animal safety, and that's it.
Yep.
So,
they're ready.
you know, think of the flu vaccine. You get it every year, and some years I get a sore arm, some years I don't. So it really depends. Did the CDC reduce the cycle threshold to be positive, Amy?
Did the CDC reduce the cycle threshold? No. What they did was they came out and looked at the data and they said so they came out with one idea, right, at the very beginning. And then they collected data and they correlated CT value to when you could isolate infectious virus and when infectious virus tailed off. And then they moved their numbers to reflect that data. So they didn't really reduce the cycle threshold to be positive. They just made it more correlative to culturing, the ability to culture infectious virus.
Great. How contagious is B1117 actually? We don't know. So if you mean contagious, how easily it goes from person to person, we don't really know. There are some, there are observations of the outbreaks, but it's not clear because when that virus went to other areas, it didn't take over. It didn't displace other isolates as readily as it did in England. So we really don't know. It's, as you will see, if you read papers, they say allegedly or thought to be more contagious because we really don't know.
Well, that's the problem with the way people interpret the data and the tests that we do.
Yes. Oh, here's for Amy. Logic says there are people from all over the world here to watch Amy. Don't feed the trolls. There you go.
That's fine.
Can you contrast J&J vaccine and Moderna Pfizer? You want to do that, Amy?
One's mRNA and one's adenovirus, right?
Yeah, Moderna Pfizer are both mRNA. It's an RNA coding for spike, which is injected into you. It's actually
they're all injected into you. They all
wrapped in a
encode for
lipid
spike.
nanoparticle, and then the J&J is an adenovirus, where the spike gene has been put in.
It's just a delivery tool. At the end of the day, they're all basically the same. You get some mRNA that encodes for a spike, it gets presented on the cell, and voila, you get some immune response.
Yeah, the J&J, of course, is one shot, which is nice, and the Moderna Pfizer are two. So that's a bit.
Well,
of
it might be
advantage.
because adenovirus is already very efficiently taken up by cells, right?
Yeah.
Whereas a lipid nanoparticle is not as efficiently taken up.
Yeah,
by cells.
adenovirus is built to be taken into cells.
Exactly.
Right?
So yeah. So.
Jeffrey, I can't agree with you more. I think journalists should not publish stories about new variants until the community has more time to study them. I agree, but they're in the business of selling news, right? And they want the news fresh off the presses. So that's what we're stuck with.
It's not necessary. Yes. So it's 50% the journalists, but it's 50% the scientists who talk to the journalists and feed them the misinformation.
Yes, right. No, we cannot say scientists are not to blame, right, Amy?
Right. I mean, there's a whole editorial by someone that miscites the literature. What are you going to,
I read an article on bacteriophages sending chemical signals to each other. Is this true? Yeah, actually, we did this paper on This Week in Microbiology a couple of years ago. So it's actually the infected cell that's sending a signal to another infected cell, and it's quite interesting. And what the signal is, I don't believe they knew, but it's true.
It's like a pheromone.
Is there guidance for whether people who are vaccinated can hang out amongst each other without a mask? So the CDC has not said yet that you can hang out without a mask after you're vaccinated. We have to wait for them to say that because we don't know the chances of transmitting to each other and passing it on. There are limited data so far, right, Amy, that vaccines may decrease transmission. One study out of Israel, Pfizer study, I think.
Yes.
But it's too early.
Yes, but I thought that they said something, but I could be wrong.
CDC?
Yeah, I thought that they had something about if you were gathered with, maybe it was that if you were vaccinated, then you didn't have to quarantine after you were with people who were exposed to SARS-CoV-2. Maybe that's what I'm thinking of.
I learned from Vincent's lecture that measles erases immune memory. Can this cure autoimmune diseases? That's a great question. I think probably not, because you can restore the immune memory that's wiped out by measles by, say, a new infection. It can wipe out your polio immune memory, but if you get a vaccine, it'll bring it back. So, the cause of the autoimmunity is probably still there. So I think it would return. What do you think, Amy?
Sounds right.
Under what conditions threshold will you roll up your sleeve for a third booster shot in a year or two from now? What about you, Amy?
If there is convincing data that we have variants that are, widely circulating, that immunity from the first shot, from the first generation of the vaccine will not protect against, then I have no problem getting a yearly booster.
We both already get boosters yearly for influenza, right, Amy?
Right.
We're okay with that. So I'm okay with this every year or two.
Yeah, I don't have a problem. You just have to show that it's really needed, right?
Yes, right.
I mean, because I got a lot of, I don't develop any antibodies against measles, and I got a lot of measles shots to the point where it became a concern about whether or not they were going to give me measles.
Does glycosylation of spike influence immunogenicity, T-cell response, antibody binding? Is
yes.
Spike of the subunits vaccine glycosylated?
Yes. So it infects all of them. Immunogenicity, T-cell response, antibody binding. There's a lot of papers that describe that. And yes, the spike in all of the vaccines that are produced by your cells are glycosylated appropriately.
Which one is better, mRNA or traditional vaccine? What do you mean by traditional? Like non-mRNA?
I guess. I don't know. I don't think you can compare.
I think it's hard to compare.
Well, yes, because you would argue that measles and the polio and Sabin are very effective vaccines, right?
Mm-hmm.
They're infectious attenuated vaccines. And when Salk is generated appropriately without the cutter episode, you would say it's also a very potent vaccine, right?
Yes.
So, and DPT, which is, inactivated toxin, right? Mm-hmm. Diphtheria toxin, which is a protein. So it's very effective, right? So to me, this is like asking if Ebola is more pathogenic than Lassa fever. Like, you can't, or virulent. You can't compare virulence. I don't think you can compare this.
But if you want to just compare efficacy, I don't think you can do that, though, right?
No, I don't think you can.
I think you take the one that's offered to you. That's what we always say. Daniel Griffin says that.
Yeah, but I didn't think that this was about whether or not I should hold out against waiting for the next vaccine. I thought that this was just a curiosity question about can you compare?
Probably it is, yeah.
Yeah, so I think it's just like we say you can't compare virulence of Ebola to Lassa, or virulence of SARS-CoV-2 to flu, so I don't think you can compare.
Do other respiratory viruses cause asymptomatic transmission? I'd surmise no, as we don't see unvaccinated suddenly getting infected left and right by those around them. Thoughts?
Yeah, rhinoviruses cause asymptomatic transmission all the time.
Yeah.
So does EV68.
Influenza
so does adenovirus. Yeah.
Yeah, they all do. There are very few viruses that do not cause asymptomatic infections.
Right. And they're all efficiently transmitted. I don't think so. I'd surmise no, as we don't see unvaccinated suddenly getting I don't know what the connection with that and the first. I don't know what the statement is, how that is connected to the question. I don't understand the train of thought. But all of those others a lot of them are asymptomatic infections. And you would even argue, a lot of gut pathogens are asymptomatic infections.
Yeah, sure. What's the deal with colchicine for COVID? So Daniel reviewed this. He said it didn't seem like, in a small test, it didn't seem like it had a huge effect. So colchicine is an anti-inflammatory, but some bigger trials are ongoing. Thank you, Faster Tortoise, for your super chat. Why are the numbers going down globally? And deaths? Is it just an effect of restrictions?
How many old people can you kill?
So I think, most of the deaths are in over 65. That's for sure. Why are they going down? So Paul Offit said vaccination and herd immunity, right?
Yeah. I mean, you started off with nothing, and now, I don't know what, 1% of the world's population has been exposed and infected?
88% of Israel has been vaccinated.
Yes, I know.
It's amazing. It's bloody amazing. Great.
Yeah.
It's really great.
It's great. And then, I mean, there was a whole article in the Times about like how they have extra Moderna vaccines that they're sending to Hungary and Latvia, and I think some places in South America. I forget where else. And then they brought it up with the Palestinian thing, and then it talks about like the Oslo Peace Accords and stuff. And somebody else already corrected me on this last week. So the
numbers going down because the variants are not picked up by PCR, Amy.
No, they're picked up by PCR. They're the spike dropout.
How about we will rock you?
No queen.
Okay. There's a rumor that some variants transmit as aerosols. It's nonsense. No data to support it. Why is the U.S. so good in vaccine development and so bad in sequencing variants? What do you think, Amy?
I think that people were given a lot of money to develop vaccines, and there was a lot of emphasis on development of vaccines. And I think the basic science and sequencing was not appreciated. And so it was not as well funded and thought to be important.
Yep.
And I think we potentially underestimated what consequences would occur when you start giving people lots of antibody therapies, and that may, having high immune pressure would would end up doing. Because these viruses were really very understudied. So we don't really understand that much about, their ability to generate escape mutants and stuff. That's what I think, but you can correct me.
I agree. They put 24 billion into vaccines and very little into sequencing and basic science. Thank you, Post Polio News, for your super chat in support of Amy's No Music. Has a study been done to investigate if people who died from COVID had received a flu shot within 6 months? No, but that data certainly could be captured, right, from the medical records if someone wanted to do it. You'd have to get a, institutional review board protocol approved to do it, but it could be good. Could be good.
Yeah, somebody could do it. We're not doing it.
Why can't we make a good human vaccine for rabies? I thought we did have a vaccine.
I thought so.
What's wrong with it, Jen?
I don't know.
Why does the Moderna vaccine use 3 times more mRNA than the Pfizer? Does it have more side effects because of it?
I don't think the side effects are from the mRNA. I think the side effects are due to that lipid.
Do you know why they have so much more RNA, Amy? It's just what they tested?
It's just what they tested, and maybe it's what they were able to package. Maybe Pfizer was not as efficient as in packaging RNA as maybe their nanoparticles are not as, RNA friendly as the ones in Moderna. I mean, it's very proprietary, right? How they do it.
Is there a collection of long COVID papers and information other than Google?
Well, they should start off with your TWIV with Fiona and Mady and everything, right?
Yeah. But there's no collection as far as I know, right, Amy? There's not a place that is collecting papers. I know that happens for other diseases, but I'm not aware of one unless someone else knows.
I'm not aware of one, but I'm not aware of a lot of papers. I'm not clear what kind of papers they're looking for. Like clinical reports? Well, no, but clinical reports or research.
Yeah.
So I don't know how we're going to, right. I don't know how we're going to ever model this exactly in an animal.
Can you explain the term fitness in regards to viruses and variants?
Ability to reproduce more efficiently.
Yeah, it's it depends where you're studying fitness. In the laboratory, when Amy infects cells in culture, she can define fitness as making a good amount of virus. In a host, fitness might be making virus or spreading from one host to another or establishing it in the population. So a variant, as viruses undergo mutations, some of the mutations are deleterious, some of them have no effect, some of them may be beneficial and they may improve the fitness, they may improve the reproduction. Another way of looking at fitness is if the mutation or the spike change, say, allows the virus to not be neutralized by antibody, that would improve its fitness in that particular host. So those are some examples. What is the best test to see if you had asymptomatic COVID a couple of months ago?
Well, the only test is looking for antibodies against viral proteins. Ian M. Nishi's paper came out earlier this month about, their peptide array.
Where did it come out? What journal?
Nature Communications.
Okay. Yeah, so you need to get an antibody test. And there are a few of those available, but they're expensive and you can't do it at home as far as I know. You have to get a prescription. If a vaccine is 95% effective, what causes the 5% of ineffectiveness?
Lots of things.
Could be that some of the hosts don't respond, right?
Yeah, for sure.
And so they're not protected. It could,
yes.
I think, the vaccine is not going to induce antibodies in 100% of the recipients because humans are all different genetically. So That's probably
Well,
the main.
infection doesn't produce antibodies in 100% of
No, the
infected
vaccination.
patients.
I mean, the vaccine.
I
Yes,
know,
infection doesn't either.
but
Yeah.
if infection does not produce if you get infected and I get infected, and you had measles and you have a measles antibody response, and I've been like inoculated or immunized or vaccinated 11 times against measles and I have no immune response.
Yep. So I think that's a main reason for that. My mother, 73, got cold sore after Pfizer shot. Can this be related? Definitely. The inflammation caused by the vaccine could trigger reactivation of herpes. We know that stress is a reactivator, so absolutely. Fever is common trigger.
Mm-hmm.
Any new information about measles, mumps, rubella vaccine and reduced COVID illness? I haven't heard anything. Not a thing. Have you, Amy?
No.
I don't know if trials are going on. I don't
I
know.
don't know why we would need a trial now. Why not just get vaccinated?
Well, you never know. If people can raise the if people can raise the money, they do it, right? Chronic Lyme versus PASC. By the way, we have a new acronym: post-acute sequelae of SARS-CoV-2 infection, PASC. That's what NIH is calling long COVID. Did you know that, Amy?
No, but it's way too long for me to say. And that doesn't roll right off the tongue. PASC, that doesn't really roll off the tongue.
Not like COVID, right?
Okay, whatever. They
don't
can
think
call
COVID,
it whatever
Rosa?
they.
Anyway, I don't know anything about chronic Lyme. I can't really contrast. Can you, Amy?
No, I don't know. I don't know how you can compare and contrast diseases like that. I don't get that question.
So Vern is in the Novavax trial. They're talking about a crossover study to get everyone vaccinated. Well, I'm not sure about Novavax's, but if the results come out and they say the vaccine is safe and efficacious, they have to offer you the option to be vaccinated, and that would terminate their long-term study. They give you the option. That's what I believe they're talking about. And some people may choose to stay in the study, and even though they have a placebo, and some people can choose to get vaccinated. That's how it works. How does it make sense from a natural selections viewpoint that a virus kills its host?
Well, it doesn't.
If it kills it after it transmits, it's okay, right, Amy? It's all about transmission to a new host.
Yeah, well, it doesn't really it's not, death is not is an inadvertent consequence.
But most viruses don't kill their hosts. So that's the
As I said, death
factor.
is an inadvertent consequence.
That's right. It's inadvertent. That's right.
It's like entering the CNS.
I was just going to say that.
It's an inadvertent consequence. All right, 2 more because it's 8:45.
I think they mean you test positive, you should wait 90 days before being vaccinated. Oh, that's an old question that came up a long time ago. Arthur, thank you for your super chat in support of music. Is it proven that mRNA vaccines prevent transmission? No, there's some early data from Pfizer in Israel that suggests it reduces shedding, but we don't know about transmission yet. It's going to be a while before we know that. Frank, thank you so much for your super chat. We appreciate it.
All right, are you 2 more questions because I'm going to go. But otherwise, if you're just going to read super chat, thank yous then.
Powers that be should ask you and Amy since this is your expertise. Yeah, but they don't. They ask other people. Let me find another question. Are the efficacy numbers as suggested by different vaccines referring to population or somehow referring to individual? It's individual. It's population. Sorry, population efficacy. Because that's how you do it. You measure 40,000 people, 20,000 placebo, 20,000 vaccine.
Then it's not really a population.
It's not individual.
You're counting individuals.
That's right. It's individuals in that study, right?
Right. It's individuals.
Denise says if you're a Prince fan, she has to work in your lab.
Yeah, I am actually a Prince fan, but I know that you don't like Prince. Sure, she can write in and work for us. Hopefully, we won't go out. We won't be broke by the time she comes.
Says CNN's reporting Moderna updated their vaccine to combat South African variant. Check your news feed. Story just broke. Okay, thank you.
Yeah, I said that earlier.
Yeah, I think you said it. All right, there's you can go if you want, Amy. I'm trying to find questions for you.
There's.
Do you see one that you want to answer?
No, I just see a lot of people who are. Why are males more prone to COVID than females? I think that's Saber Klein. Saber Klein should answer that question. Then there's some snarkiness.
Disappointed you did super chats. We're doing it. Well, someone asked me to do it, frankly, and I will use the money to support our.
To support you.
Like Jeffrey says, I'm happy to support your work. I do answer all the questions. I don't like.
He raises money for a living and enjoy being on the other side of the equation. So, Jeffrey, you can help Vincent raise money. He's not good at it. He's not a good money person. Here's one. Does cross-reactivity between SARS and human epitopes explain long COVID? No. It's from this AP Med person. No, they do not. It does not explain long COVID. Long COVID is a condition related to the immune response, like the cytokines and chemokines that are released and the damage that they do. At least that's my understanding.
I think that's correct.
And somebody wants to know if I have ideas why I don't generate an immune response to measles. Seems very specific since you're not a bubble girl. I have a few ideas of why I don't generate immune response to measles. I am not a bubble girl. That is correct. However, there's no way to really know. I'd have to do a lot of sequencing and stuff. About why I don't generate. I think like there's a defect in class switching and stuff for some reason. But it also may be because, there's also, people have different point mutations or SNPs and other genes that help develop an immune response. And I might have a point mutation in one of those genes that is specific for measles. Like IRF7 by Casanova, specific against flu, but not rhinovirus, right?
Mm-hmm.
So one of those genes might be defective, for measles, but we don't investigate that because the people like me are probably very, very rare. Right?
Yep.
And my focus is on EV68, not measles. All right. Now it is time. Yeah, now it's time for me to go. We're going to go pick up the sequencer tomorrow, right?
All right. Yep. I'll be there. See you.
All right. Good night. Have a good evening.
Bye. All right. Now you got me. Let me go back up here and answer some of these older questions. Let's see. Post polio. Super chat from Frank. Yes, I remember that. Super sticker from Susan. Let's see. Some people are mentioning the New York strain or nut strain variant. Thank you, Jeffrey, for your super chat. There is a new variant in New York City, which was found in November, and they say it's spreading. It has spike changes that could affect vaccine efficacy. And this is, again, the newspaper is publishing this before any preprint even is out. There's no preprint out yet. So I think it's kind of irresponsible. And, you know, the writers at the Times and other places know who you are. Thank you, Tom, for your super chat. And thank you, Susan, for your super chat. Are there more studies of long COVID? No, the NIH is getting them started now. People are starting to take this very seriously. So, but it's not going to happen overnight, right? Where should one go to get the know on cat viruses? A veterinarian, right? A veterinary virologist. I can find you some if you'd like. Just shoot me an email. Yes, Susan. So that's the article from Columbia. So it's a New York Times article. It's another variant. It's been around since November. It's happening. However, the issue here is the numbers are going down. So these are becoming less and less relevant. We are tracking them, and that's good. Totally good that we're tracking them. Yeah, phase 3 testing of under 18s is ongoing. That's good also. What's the likelihood of universal corona and or universal influenza vaccines? I think it's pretty good. I'm not, over 50% chance of them happening. The influenza are in phase 1. Corona is still under development. I would say the influenza maybe in the next 10 years and probably the same time frame for the corona. I think we're going to have universals for both, yes. Do you know why a person didn't get symptomatic with significant malaise until 4 days after the first Pfizer? So most people, react within 24 hours. I would say you probably have a suboptimal immune system, right? That's just taking longer. So if you have ME/CFS, maybe that's part of it. I don't know, though. That's just an idea. What protects children from severe disease? So some children do get severe disease, multisystem inflammatory disease. It's rare, but it happens. Why children do not get severe disease is being investigated. People are looking at their immune signatures to find out. I think we're going to learn a lot about that, but right now, we don't know. What does E and RDRP2 mean on the PCR test? So the E is the E gene primer. So you have a separate cycle threshold for 2 primer sets. So your PCR had an E gene primer set and an RNA polymerase, RNA-dependent RNA polymerase primer set, and the CTs were 18 and 21. So that's pretty low. It's quite a bit of virus you have there, and both of them are close. They're kind of confirming each other. Okay, Raphael. Thumbs up, please. Yes, if you have a chance, give us a thumbs up. What do you make of the major decline in cases in South Africa? I think a lot of people have been infected. I think, remember, the 70% herd immunity to stop transmission is based on an R0 of 3. If we're reducing the R0 by interventions, then you'd need less at that particular time. So I think that in vaccination, well, in South Africa, I'm not sure how many vaccines they've distributed. But I think globally, this is a major effect of infections. I think the same is globally. Wherever there are declines, it is because of the number of infections. Dr. Chu says the devil is already here. So Chu, I've known for years. He's a genomics guy. That's what he knows. End of story. It's not helpful, but as you mentioned last time, Jeffrey, people want their 15 minutes of fame. They love getting in the New York Times. These efficacy percentages refer to stopping symptoms. So it's severe COVID, yeah, severe, mild, moderate COVID. Not a question. Thank you for your virology lecture series. You're welcome. I'm just about to post after this the 13th lecture tonight. I love doing it, so my pleasure. What would you say to someone who is too scared to take the vaccines? One of my friends took it and she was crying. I mean, there doesn't seem to be reason to worry. Well, I would say that, I would ask them why they're scared and try and reason with that. And then you could tell them that many millions of people have received these vaccines, including the host of This Week in Virology. He's gotten 2 doses and he's okay. And it's fine. I know a lot of people who are getting them. Of course, it's hard to reason with fear sometimes. I totally understand that. But I would try and ask them why and point out that a lot of people are getting them. What's the risk of getting SARS-CoV-2 while lifting weights in a gym with more than 2 to 3 meters separation and everyone wearing masks? So I know that gyms are letting people back. In fact, Amy is running at her gym now and with masks. So far, I haven't heard of any outbreaks. So I think it's low as long as everyone is distancing and keeping mask wearing. That's the important part. Cells at work. I have it here on my shelf. Here. My son gave it to me for the holidays a couple of years ago. I love it. It's fantastic. Actually, this is the manga. I'm sorry. So I have the manga here, but the anime, haven't seen the anime. I have to take a look. Is it on like Funimation or something like that? What do I think of ivermectin? So in the beginning of this outbreak, ivermectin was shown to inhibit the virus reproduction in cell culture. It was very interesting. And, ivermectin is an approved drug for river blindness and other parasite infections. So it was thought it could be used for COVID. And a few trials have been done, small trials, that show some moderate effect. And Daniel thinks it's not because it's inhibiting virus, because it apparently has no effect on viral load in patients. So maybe it's inhibiting something else, maybe secondary infections. We don't know yet. But Daniel is okay with using it. Some studies say that in Brazil, some cities have 70% immunity. Yes, I don't understand that because these cities like Manaus are just had another outbreak. So it doesn't make sense in terms of high immunity. Something is wrong there. That maybe the number is wrong, the calculation was wrong, and so forth. That's something we talked about on TWIV last week, I think, and it needs to be sorted out still. Does the Baltimore classification imply a phylogenetic relationship between viruses? No. The Baltimore scheme is just an informal way to classify viruses by genome type that helps us to study them. But it's not a formal taxonomic arrangement, okay? And viruses are polyphyletic. They seem to have multiple origins, right? A long time ago. We don't really know because we don't have any really old specimens. So it's all implied by evolutionary clocks and that sort of thing. But the Baltimore scheme implies nothing about that. Is the way the MHC presents virus fragments on the antigen-presenting cell different depending on what vaccine platform is being used? So the antigen-presenting cell will pick it up. Will pick up the protein and process it and put it up on MHC class 2. The protein will be endocytosed. Now, that's an endogenous presentation. The protein is picked up by the exogenous system, and it's endocytosed and processed. So there are 2 different ways of presenting. Do we know of any past vaccines that demonstrated safety upon initial release but were later shown to cause issues years or decades later? You know, I asked Paul Offit that. He's the vaccine expert on last Friday's TWIV. And so here is the examples. So the rotavirus vaccine, the first rotavirus vaccine, a few years after its release, was associated with a slight increase in intestinal intussusception, where the intestine telescopes on itself, which is caused by the natural rotavirus infection also. So that vaccine was withdrawn. It was replaced with one that doesn't apparently cause that. Poliovirus vaccine, actually very shortly after its licensing, was shown to, the oral polio vaccine was shown to cause polio in 1.5 million kids. And we're still using that vaccine. Those are the only two examples. And Offit said all the others, if you go a few months, there's nothing else that happens in the long term that he's worried about. Can you say that the variants are no less deadly and no less contagious? Trying sidestep, mistaking absence of evidence for evidence of absence. So I want the evidence that if you're going to say something goes through the air better in droplets, you better provide it. Otherwise, you scare people. These viruses are more fit. They displace other viruses. That's why they penetrate through the population. It doesn't mean they're more droplet transmissible. If one has and gets an mRNA vaccine, will they have to keep with mRNA-style vaccines for the rest of their lives? No, not at all. No, for sure not. I mean, next year I'm going to get a flu vaccine. It's not going to be an mRNA vaccine, even though I got mRNA vaccines. You may be able to mix them. Even for COVID, you may be able to mix mRNA and adenovirus. The trials to check that are ongoing now, so we'll see. What do you think about long haulers only getting a single vaccine dose? Why would you do that? Are you worried about? I'm not sure why that would be an issue, because I'm not sure that one dose is enough to protect, right? Although a long hauler has now recovered from infection, so it should have sufficient immunity boosted by the vaccine. So this is what people are thinking. I'm not sure it's official policy yet. What do I think about it? I don't know until I see the data. I want to see the immunity data. Can we eradicate SARS-CoV-2 like we did with smallpox? I would say no, because I think it's gone into animals in the wild. It's gone into mink. It goes into cats. I think it's in rodents, and they're all over the place, and we just don't know it yet because we haven't checked. So in that case, it'll be coming back into people from those sources. So I don't think we can eradicate it. No, it's here to stay. Any more data on the severity of the UK variant? I look every week and I don't see it yet. Very hard to do. It's all going to be an association where they look at people who have been infected with the variants and with the previous isolates and try and compare what kind of disease they got. And a preliminary study a couple of weeks ago suggested that the variant might be associated with a higher mortality. But they agreed that the data were not sufficient. I haven't seen anything new since then. But every week when you come here, I'll update you. Have either of you been antibody tested? I have not been. My doctor's office got Moderna and claimed the doctor did test antibody positive. Again, the Moderna is going to give you antibodies to spike only. So if you do get an antibody test, make sure it's testing for spike antibodies and not because there are other tests for antibodies to other virus proteins, right? And the vaccines do not give you those antibodies. Yeah, they're saying the California variant is so severe. I don't see the data for that yet. They're jumping the gun. I think this is so irresponsible, and this is what happens in a pandemic. And even when I see a preprint, like the UK data, it wasn't even a preprint. It was just a study released. I wasn't convinced that it was more virulent. I mean, I don't have any horse in this race. I just want what's real, and that's how I'm seeing it. If spike isn't modified to lock in pre-fusion, does it tend to assume post-fusion? Some other predictable configuration or some not very predictable? No, it assumes, so if it is properly cleaved, it will, which it would be in the cell in which it's made, it'll, or even if it's not cleaved, it will fold to a certain frequency to the post-fusion conformation, and it'll be less stable and expose fewer neutralizing epitopes. That's why you want to put it in the pre-fusion. Although the ChAdOx, the Oxford AstraZeneca vaccine, is not pre-fusion locked. So, I don't know if that's related to the little bit lower efficacy of that vaccine. It's very interesting. I've recently learned about the concept of original antigenic sin. Will this make future boosters less effective? Could a second vaccine dose somehow imprint the original strain more than a single dose? So original antigenic sin, is typical with influenza virus, that first virus you were infected with, whatever you're subsequently infected with or vaccinated against, you tend to make an immune response to that first isolate. So that's kind of problematic. It dominates your immune response because you want to be dominating to the current vaccine that you're getting, right? So would that happen with SARS-CoV-2? We have no idea because these variants are all new, right? We just have to see, and we have to vaccinate people. If we make variant-based vaccines, we have to vaccinate people and check their antibodies to see if they're biased towards their first vaccine or that one. Don't know. People are fairly serious about masking in California. That's good. I'm glad to hear it because in New York City, they're pretty good too. What do you think about the idea that the mRNA vaccine could create anti-syncytial antibodies? I don't think there's anything to that. There's a very short homology between syncytial, which is a fusion protein, and other fusion proteins, right? Like the spike fusion protein. And I don't think it's enough to make any homology. There's no evidence that this would be a problem. So this has been floating around for a while, but it's not really, I don't think it's really an issue. Thank you, Faster Tortoise 2, for your super chat. Are there any signs the world will learn something from this pandemic, or will we repeat the whole mess decades from now? Well, I'm not going to be around to find out. I don't know. So we're going to have another flu pandemic probably in 10 or 20 years. Coronavirus has been every decade so far, although not of this magnitude, right? But they are going to increase in frequency as we encroach upon wildlife more and more. I would like to think we've learned a lesson, but I think there is generational amnesia, frankly. I think once this gets far enough in the past, people forget. I mean, we could have prevented this pandemic, but we weren't ready because we didn't do the right research. So I'm hoping we'll learn from it. I really do hope, but I don't have a lot of faith personally. I don't know anybody in real life that are getting the vaccine other than my kids and dad, and I think that's because I've had Vincent on TV hours a day for a year. Well, I'm happy to have done something good. Everybody I know is getting the vaccine, and there are people I know who haven't been able to get it, and they want it. It's different circles, I suppose, right? I mean, my wife got it, and she wants to get it for our kids, but they're in their 20s, and they can't get it right now here in New Jersey. But we want to. Having an autoimmune disease increase your chances of having a cytokine storm. I've never seen that association, Belinda. I'm not aware of that. Traditional. A lot of media sources call the viral vector vaccines traditional. It's funny. Vectored vaccines are pretty new. The media doesn't know anything. Traditional vaccine is an inactivated vaccine like the Salk polio vaccine, or an attenuated vaccine like the polio, Sabin vaccine, or the measles, mumps, rubella vaccines. Those are traditional. Vectors are really new, major media, come on, get with it. Take a virology course. Hey, I know which one you could take, by the way. Virology.ws/course. But they'll never watch my lectures. Are you serious? It's too much work. They want to write articles and make a living. Sorry, I'm ranting a bit. So, yeah, that's my feeling about that. How do I feel about opening schools? Portugal had 16,000 new cases per day less than a month ago, and now it's less than 1,000 thanks to lockdown. Starting to discuss schools, though. So I think if all the teachers are vaccinated and everyone's wearing a mask, you can open the schools if you have low case numbers like you have there. I think it's okay. U.S. numbers dropped surprisingly fast. Actually, the numbers have only dropped a few weeks ago, right? In January, we started to ramp up the vaccine production. But I do think that the outbreak, the big bump after the holidays, really immunized a lot of people. And maybe a lot of people started getting it and wearing masks, too. Will climate change bring more novel viral epidemics as vector habitat ranges bring more frequent contact of new species with more humans? Yes. Warming will increase mosquito ranges, right? And they carry a number of viruses, so they'll increase, and we will contact other animal species that have, and our industrialization and, repurposing land puts us in contact with other species. So I think the numbers are going to keep going up unless we're ready with universal antivirals and vaccines. We can be ready. It's just a matter of spending money. So you can spend $2 trillion on recovery, or you could spend, $5 billion on universal vaccines and antivirals. It's an easy choice, I think. Thank you, Kaliap, for your super chat. You think I need some moderators, perhaps? What properties make measles so highly transmissible? Because we've lost 200 people already. I think the virus can travel in the very tiny droplets that you produce that travel hundreds of feet. Because you don't need a lot of virus to infect someone, and those droplets don't have a lot of virus in them. And so I think that virus can produce a lot in the tract and gets into those smaller droplets and goes a long way. But I don't know. That's speculation. I don't think we have any data on it. Is there any truth to the idea that the more we vaccinate, this will put positive pressure on the virus to mutate? Well, no, because we're dropping infections at the same time, right? So the way you get variants is by having more people infected. We saw that from November on. We had huge infection numbers and a lot of variants emerging. So if we knock down the variants, we're going to push down the fodder for the selection. So I don't think that's an issue. Maybe if you called it fundamental instead of basic science. I have heard that suggestion before, for sure. Hey, we should call it fundamental. I agree with that. I keep forgetting, though. When the lockdown goes away, do you think people will be more vulnerable? Thank you, Tom, for your super chat, to cold and other viruses because we have socially distanced and lowered our immune system. No, I don't think so. It's a good idea, but I think we have plenty of immune system educating anyway. So I don't think that's going to be an issue. Ruby's Red, thank you so much for your super chat. You like the basics of infection. I thought that was a good one, too. A lot of people didn't watch it, though. More RNA used by Moderna because the transfection efficiency isn't as good. Maybe the lipid nanoparticle isn't taken up as efficiently. It's possible. I mean, all these things could be studied if you want to, right? Why was SARS-CoV-1 able to travel through aerosols and still infect, but SARS-CoV-2 cannot? I'm not sure that that's correct. SARS-1 isn't shed until the peak of illness, and then you're in the hospital. So that's why we were able to contain that. And I think some of the longer distance aerosol Transmissions were fecal plume sorts of issues in the apartment complex with the faulty plumbing, right? I think they happened rarely, but was not the norm. Oh man, I remember this. TWIV needs more cowbell. That was from something. It's so long ago, I forgot. TWIV needs more cowbell. That was an arc at one point. What happened to the Mina home test? Never got made. Don't know why. I don't know why. So many people have saying that the cases are going down because guidelines have for calling positives was changed. No, that's not true. As Amy mentioned before, that's not true. That's not what they did. Yes, PLOS Pathogens has a COVID-19 research collection, so you could look at that for long COVID papers, right? Since we're getting good at SARS-CoV-2 vaccines, might we use this tech to develop a vaccine against the common cold? For sure, definitely. And other viruses, right? I know that Moderna had some herpes virus vaccines and HIV vaccine mRNA candidates. So now, given the success of this, I think we'll see more. Absolutely. Absolutely. So your old TWIV Nipah virus is the local word for tropical fern, abundant in the countryside. That's interesting. So I was in Singapore in December 2019 for a Nipah virus meeting. I thought that was the name of an area where it first emerged, but I believe you, a fern. I'm avoiding the trollers. You know, you guys have educated me. I know someone who was scheduled for second shot on March 2nd, supposed to start chemo on March 8th. I think that's too close. I would mention that. I would mention that to the docs and say, you know, I want to get through the generation of antibody first. Are mRNA vaccines the way forward? So they might be for some others too, but we'd have to test them. I don't think they're going to work for everything. We just have to see. We have to test new ones. Can the vaccines be mixed? They're doing a trial to figure that out right now. We just mentioned it on Tuesday's TWIV. There's a trial to check Pfizer and AstraZeneca in different combinations to see if this actually is what's happening. Long COVID names. Yep. Oh, now I just got to Amy's question. Does Amy have any idea why she doesn't generate an immune response? No, she suggested she may have some problems with class switching and so forth, but she really doesn't know. How does immunity work in the first week? After my body clears the virus, I can't get reinfected right away. This doesn't happen. Well, you get infected and the virus is being sampled by your immune system for 2 weeks, and the antigen probably stays around for a while. But then the educated cells do their own thing and hypermutate and continue to get higher affinity antibodies. The male-female serious disease difference is really interesting. Over time, will COVID-19 be like the common cold and become mild? If so, how long will it take? So I think Ralph Barrick last year suggested that it would become a common cold virus. And he said, it could take 5 years or 50 years. We don't know. I think the current coronavirus, common cold coronavirus, has probably emerged many years ago as a pandemic, a serious pandemic. So it's possible this will do the same. It's going to just take time to get global immunity. I think that's what ends up making these infections more mild. How does government work in infectious disease differ from academic work? In some ways, it's very similar. The structures are different. I have my lab, and Amy has her lab, and we do what we want. And in the government, you're usually reporting to someone who tells you what to do, although you do have some flexibility. But, in the end, you're both trying to solve problems. How is it determined some of the vaccines are tested against 2 shots? Thank you, T. Klupper, for your super chat. And some only 1 or 2. It is just how they plan the trial to begin with. So they may have had some animal data, preclinical animal data in the beginning, which said, 2 shots are the only thing that's going to work. So they went with that in people, and J&J had data saying that 1 would be enough. So I think that's how you get informed, and that's how you move forward. I do need a moderator for the future. Here's Tom's super chat. Thank you for your super chat, Tom. Rubies are red. Thank you, super chat. Basics of infection. I have good graphics. It's from the textbook. What advice do you have for someone with a degree in science but no work experience get their foot in the door? Go volunteer in someone's lab. And say, I will do work in your lab and not get paid, and then show a lot of enthusiasm. And then when they see you are enthusiastic, they'll hire you, and they get you to start doing some experiments. SARS-CoV-2 variant question. Could there be RNA tertiary structure stretches of nucleotides that could make the polymerase selectively make specific variants? This is one of the ideas, that the recombination is occurring in certain places, and that's causing these deletions in some of the variants, and that would be a property of the polymerase. Yes, that's absolutely being looked at. Really interesting idea. Thank you, T. Klupper, for your super chat. I appreciate it. Can we get infected naturally from a vaccinated person who's making spike? No. The spike doesn't transmit from person to person. Okay, Frank, get in touch with me. You can help moderate. The next time we'll set up a way to do that. I'm taking vitamin D3 daily, yes. I've been doing that since the winter, when a physician said you should anyway in the winter when you're not getting much sunlight. Don't worry, Eddie, about super chats. It's not necessary. I like you all. And don't tell me I don't understand certain things, because you have no idea. Other people, you have no idea what I do or do not understand. Can a moderator get rid of the trolls? I think so. There are ways to, you can see people who are making dumb comments and provoking, and you can just shut them out. Yeah, Frank will do that. He's a tough guy from New York City. We have 7 minutes left, folks. Does the J&J 1 dose have the same efficacy of the 2 dose? No. It's not in the 90s. I don't remember the number now, but it's not as good, but it's really good. And, sometimes 1 dose, offsets the thing. I'm a grandmother who wants to visit my grandchildren. I received my 2nd vaccine 3 days ago. So I would wait 4 weeks in your case, and then wear a mask and go visit them. I think that's good. You'll be okay. But you're going to have to wear a mask to protect them, right? Because they're not vaccinated. Can you explain the difference between more virulent and more fit for a layman? What makes a variant replace another without being more virulent? So virulence is not needed for viral fitness, right? As Amy said, virulence is a side effect of a virus reproducing in the host. And all it needs to do is reproduce effectively and transmit to a new host. And if things change that, then that's selected for. A virulent may be a side effect. So let's say to transmit to a new host, you have to replicate the higher titers. So that may end up causing more disease in the host as a secondary effect. It's not a primary effect of the change. So virulent is disease. Fitness is just overall reproductive capacity and the ability to spread in a population. And sometimes the virulence will come as a side effect. But remember, most viruses in their hosts are not virulent. They are not causing any disease at all. It's a nightmare pandemic with very high death rate, more likely because our incursion into the natural world. So we have a high death rate here, I think. I mean, even if it's just a low percentage of the population, it's a lot of people dying. Many more people died in the 1918 pandemic, but I think that's because we had poor medical care in part at the time. I think the key is transmission, right? So you don't have to have a high death rate to get high transmission, and so that may be enough. And if you have a 1% death rate and everyone's infected, that's a big issue. It's a big issue. Do any of the pan-vaccine platforms seem like they would be viable for immunity against multiple strains? So the pan-vaccine platforms are, well, there's influenza where they're doing the spike, the stem of the hemagglutinin, and you could express that in an mRNA platform. It could be vectored. You could even use it in influenza virus. So, I mean, that's the whole point about pan vaccine, is that you hope it will neutralize any variant that arises. The variants are arising elsewhere in the stem, and the conserved residues are elsewhere. Thank you, Susan, for your super chat. We have no data on whether the vaccines prevent transmissibility. We will have it soon, though. There's Susan's super chat. Thank you so much. Greetings, Boston College. Nice to see you. How do viruses help us? Well, hard to know with humans. We have lots of viruses in us that reproduce regularly and don't cause disease. So the idea is they must be beneficial, like our microbiome is beneficial. In animals, we have evidence that viruses can help animals. And if you take my lectures, you can see that. Go onto virology or YouTube. Right here on YouTube, there are my virology lectures, and you can see some of the evidence for that. What do researchers mean when they say wild type in regards to viruses? So, wild type simply means a variant or a strain that you're using to compare with. So here with SARS-CoV-2, the Wuhan isolate in 2019 is considered the wild type because we compare everything to that. That's all it means. It doesn't mean necessarily from the wild, although that's a clinical isolate originally, right? It's just a term for saying this is what we're comparing everything to. And in fact, for many viruses, they're grown in the laboratory for years and not really related to anything that's in the wild anymore, but that's just a term that we use. How do you determine vaccine dosage strength? Well, you typically do a dosing scheme in the phase 1. You can give a few doses to different groups of people and measure immune responses and decide. And that's what was done with some of these vaccines as well, deciding which dose you can give them. Are viruses mutagenic? Sometimes the retroviruses that make a DNA copy of their genome and integrate into the host, this can sometimes be mutagenic, and we have multiple examples of that because it integrates next to, say, an oncogene, and it turns it on. Yes, Frank P. is going to moderate. He's going to get in touch with me, for example. Do some viruses have DNA and others RNA? Does that make a difference? Yep, that's absolutely right. There are a lot of DNA viruses like herpes viruses, pox viruses, adenoviruses, and a lot of RNA viruses. And does that make a difference? Well, the RNA viruses mutate more frequently. So you can see SARS-CoV-2 is mutating quite frequently here. The DNA viruses tend to less. They have a lot of other differences as well, but they both exist, and they both coexist in the wild. Well, folks, that does it for tonight's live stream. I hope you have enjoyed it. And we'll see you next week, same time, same place. Thank you all for participating. Thanks for your likes. Thanks for your super chats, super stickers. Really appreciate it. Like the conversation, and I hope you enjoy this little community who's interested in viruses here. See you next time. Good night.